Yeah, good evening, everyone. Welcome to this second and final live session for the course Functional Genomics. Um, I hope you have been going through the lectures and finding it useful. And those who will be going for the exams, all the very best. For those who have joined here for learning the subjects, I'm sure you found it interesting and useful. Um, we are looking forward to uh, questions from any of you who have joined this live session with regard to the topics that we have discussed. And of course, there are many queries that are there in the discussion forum and that are being addressed. We need to also understand while understanding or the question or understanding the answers that there are for everything there is multiple possibilities, but not all possibilities are really that is workable solution. So when you really look at any technique or the approach or the methods that we use, the methods are something that is reliable, which you can reproduce, you can repeat, and more often than not, they are successful. So that's how the methods are standardized. But there are certain exceptions, there are certain remote possibilities, or one of the several steps, this particular method being, that is not considered as something, robust approach. So it's like if I have to, my, I am in destination A and I have to go to destination B, which is say about, 15 meters away. The best way is to walk, although it is slow process. So that's what we call as approach. But if it is 10 kilometers, then one can go use a two-wheeler or a motorbike or a car, which takes you faster. Walking may take much time. So that's what the approach is, right? So that's what we really look at in any of the scientific, research. We look at appropriate method, which is likely to give you an answer for the question that you're asking. Having said that, those who are watching, you may, if you have any query or question, you may post them in the chat box that is there in the YouTube channel. So I'll try to address some of them. Um, there's a question about, it says that, let me read it out, that what does it mean HG38 with regarding the reference sequence and how can we generate the reference sequence for alignment? When we refer to the reference sequence, these are reference sequence that are normally that are there in the NCBI, database. So for every gene, so you may have multiple sequences that are submitted. Mostly reference sequence refer to the full-length messenger RNA sequence, which what we call as a copied DNA, which you sequence it, and that tells you what's the coding sequence. And when you have a multiple clones from multiple labs that are sequenced, then you're going to have multiple entry. So what the curators in the NCBI database do is to analyze all these sequence and they generate a standard sequence for every gene. That is what called as reference sequence. And I don't know the whatever HG38 maybe refers to a particular sequence, but that is what reference sequence. Reference sequence is one sequence and that is curated full length sequence. It may be, you know, um, the sequence need not be from a single clone. You have to understand that because the mRNA, you don't always get a full length mRNA or full length cDNA. But then you have multiple cDNAs, they curate and then they give the longest possible cDNA sequence for a given gene. And that is what you call as a reference sequence in terms of the coding sequence. So that will have certain unique ID, which you can use it for a variety of purposes, even when you do the nucleotide sequence analysis, alignment, or you can translate and get the protein out of it and then do the analysis for that. Or if you identify an mutation in a gene, the way you number the nucleotide is based on the reference sequence that is always considered as the master sequence, right? And that's the answers roughly to your question. There's another question: what is a consensus sequence? Say, for example, if you're looking at promoter element, right? So every gene has got a promoter onto which a given transcription factor comes and bind, like Tata binding protein, like transcription factor 3 and so on. Now, the same transcription factor will bind to a number of promoters, right? Number of gene promoters, rather. So when it does so, so if you really identify the region where a given transcription factor binds, and if you compare all these sequences, where it binds, you will find that there are certain sequences that are common, right? So that is what is called as consensus sequence. That then they sort of after this kind of sequencing and alignment, they show that this is the typical structure, consensus sequence for a promoter onto which this particular transcription factor will come and bind to. And in future, if you identify a new gene and characterize the promoter, and you want to quickly check what is the transcription factor that might bind here, then you can do this promoter characterization. That time these consensus sequences are used to identify the promoter. So that's what it is. Yeah, because there are certain residues that are uh, invariant, meaning invariably present. There are certain sequences which may be one of the two bases. So there are letter codes for that, and that's how they arrive at consensus sequence here. Well, others can likewise add your queries, and we'll be able to answer them. There's a question that is, if a gene is responsible for a species' adaptability to the environment in which it lives, how does it work? If a gene makes protein, then how does it lead to the ability to survive in the environment? You need to understand one thing, that uh, any living organism that are naturally present, wild species, all these are a result of mm, evolution. This is not something that is made. For example, it's not that um, your genome is coded and created the way we have, and that led to what we are now as Homo sapiens. So these are evolutionary forces, right? And when I say these are evolutionary forces, what do they mean? Say, for example, there are regions within a country that is very cold, or very hot, or very humid, or it could be very dry, and not every species can survive in each one of these, all the zones. So there is something called as adaptation, right? If it is very cold, then there should be some ability in the physiological pathway that helps that species to survive in that region. Likewise, it's a very dry area, then there has to be certain things. For example, we need to live with very, very little water, right? Or it is extremely humid and a rainy area, then you have a different set of challenges there. Now, the question you are asking is how these genes, help the species to adapt there. It is the other way around. So what happens is that when species migrate from one place to the other, which could happen because there is some change that happened in that area that they are no longer able to live, then they have to move. When they move to a new area, not every member of a given species, or not all species, are able to live in that area, because it could be a new challenge there. And only those members or species that have a gene variant that help them to survive, they survive. So it is simply a matter of chance that there were some variants that are there that probably did not confer any uh, advantage to that species in the previous location. But because of some change, they moved to something else, or that area there is a change now, environmental change. Now this variation really helped that member to survive. And since that member survived, and there could be other such members who had very similar kind of a gene change now that made them to survive better. Now the rest are disappearing, and they come together and breed. Now every member of this particular clan or group would have the variant that confer them the advantage, right? So that is how you will find that it is helping. For example, there are freshwater fish which live in the lake, for example, and the lakes is not that lake would have water all through the year. And in peak summer, there may be very little water or almost no water. So they dig and then get in there and they get into kind of hibernation. Likewise, it happens in cold temperature and so on. So that is so these are they were having that kind of variation. They're able to survive. So it is a selection of certain members of a species or certain species that had the ability to survive. They were selected, and after that, they survived there. So therefore, you find all the members of that species having that features. That's what it is. So it is as you see, this is first a randomness, and then there is a selection, and after that, you find a kind of a uniformity. That's what it is. Yeah. There's a question on difference between basal transcription factor and activation. So for many uh, genes, there is a steady state level of transcription, meaning the amount of RNA that is made that is kept at certain level, right? But at times we may need more of it, right? For example, there are many, there are many transcription factors that regulate stress response pathways. For example, you are experiencing a heat shock, right? So, but then there is a transcription factor called as heat shock factor one. Yeah. So this is the one that activates all the genes that's involved in heat shock response, which makes this heat shock protein, which is very critical for cell survival in conditions like very high temperature or low temperature and so on. Now, it's not that heat shock factor is not expressed or that protein is not being made. So there's a steady state level, right? For that, there is all these genes that are regulated by the HSF1 that they are being made. Now, when there is a stress, then the transcription level of these targets of the heat shock factor 1 goes up. So that is what induction. So either it could happen, it can happen at two levels. One, the transcription factor is at a stage where it is not overactive, meaning it is in a very basal condition. But whenever there is a change in the temperature, then it undergoes some post translational modification. Therefore, it becomes an activated form. Therefore, it is able to bind more often and produce more mRNA. So that's these are the two different states. In some, it could be an activator. There is a basal transcription factor that comes and binds and makes the mRNA, but then there is a slow process. But then there is an activator that comes and binds due to some signaling mechanism. The rate of the transcription goes up. And that is for a specific period. Once that the need is taken care, then again it will come back to the basal level. Yeah. Yeah, if there are other questions you may want to write there, we'll discuss. Yeah. I mean, you need to understand in transcription there are a variety of processes by which it is regulated. It is not only the amount of RNA that is being made, even the stability of the RNA, like how long a given RNA would survive in the cell could be modulated. Some of our RNAs are degraded very fast, so the amount of polypeptide that mRNA can make would go down. And there are also regulation at translation level, right? You have an mRNA, how fast the peptide should be made, whether the translation still should be halted because of certain changes. These are the other levels of regulation. So it's a complex process. There are a variety of examples over there. I do not see any more questions. We'll wait for maybe a minute, and if there are no questions, we will close this session and we'll go on. Well, it says, okay, I did not say that yeah, change in the environment will. There's a question. It is a follow up to the earlier question that a species living in hot environment migrates to humid area. It will lead to change in the gene. Well, I did not mean that. I mean, I don't think the environment can change. That's called as a mutation. Mutation could be often harmful, right? What I meant was, you know, when when there is meiosis, when you make gametes, whether it's sperm or egg, during this process, there are, the copying of the DNA takes place, right? That DNA is nothing but carrying your genes. Now, in this process, there could be error, right? So there could be some change in the nucleotide sequence, or it could be duplication of the gene, and so on. Now, some of these changes would affect the gene function. Therefore, if that particular sperm or egg that's carrying the mutation fertilizes, that individual would have some deformity because of the mutation. That is eliminated from the population because that individual does not live long enough to pass it on to the next generation. So most of the abnormal mutations which affects the survival is eliminated. But sometimes a change need not affect your normal function. So you are able to live. It is simply like you having, instead of 5 fingers, you have got one more finger, say sixth finger. The sixth finger is not going to affect your survival. So you are as good as anyone who is having 5 fingers. Only it looks a little different. That's it. But it does not change your survival. That's exactly with some of the mutations. It doesn't affect your survival, and as long as you are in that environment. Now, for example, a change in the nucleotide has conferred something new property. Let's assume we have hemoglobin. The hemoglobin has certain affinity for oxygen because that carries oxygen. Now, let's say you have had a mutation in the germ cell that made you. As a result, you have higher affinity for oxygen in the hemoglobin. Now, what will happen? In the same environment, you're going to be happy because, after all, it helps you to get more oxygen, so you're as good as anyone else. Well, let's assume that you, along with your friends who are not having this variation, um, travel and trek to a higher altitude in a mountain, and then immediately you'll find a difference because as you go up, the oxygen level goes down. So your friends who are not having this mutation will find it difficult because they are getting lesser level of oxygen in their blood because there's hypoxia, there is a lesser oxygen in the atmosphere. Whereas you had an mutation, therefore your hemoglobin has got higher affinity for oxygen. Therefore, even the less level that is available outside, you are able to capture efficiently. So you are much more, for example, um, agile. You are able to move faster than the others. So therefore, this change in the nucleotide, which gave you an advantage of surviving in a lower level oxygen, atmospheric condition is beneficial only when the environment changed. Otherwise, you had no difference like any others, right? Now, if that is the case, like, you're able to survive in a condition others could not survive. So that is how it is, right? That is exactly how, you know, a, a fitness value, which came in not by design, by error, is selected because if there is a change in the environment and if it is beneficial, it is selected. And then every member of that, species or group will carry that because you're able to survive in that condition. So that's how it comes in. So it's not that the environment makes the change in the gene. It is a change in the gene happens without any knowledge as to how it can help you or help otherwise, but it is selected depending on the change that happens in the environment. For some, there may be some who had a higher affinity for hemoglobin but lived in plains. They never found it helpful because they are just like anyone else because oxygen level is high here. But you find a difference only when you move up. So that's what it is. There are some variants that are selected when the environment change. And that is by chance, it's not by design. Nobody picked up saying that you had this ability, therefore you can go up. It's not the case. Yeah, SNP variation SNP is from parents to offspring. Yeah, I mean, any variation that you have a majority or almost 99.9% of them are inherited from your parents. Rarely variations come in you during cell division or germ cell differentiation. So it doesn't happen. It's a very rare event. So majority of the SNP that you have inherited from your parents. So easily you can trace that, that which variation came from which parent and so on. Yeah. I guess that's all for today. And thank you everyone for joining and wish you all um, the best for the exams and so on. Maybe later for some other course somewhere you may meet. Until then, take care. Goodbye.
Thank you so much, sir. We will end the
Thank
session
you.
now.
Yeah, thank
Yes,
you.
Vijay.